Hello, everybody. It's Michael Pierce here, back with Ilya Dubovoy. It's part two. You know what's going on. We have a very complicated graphic on the screen. Already has me excited. Ilya, continue. Take it away.
All right, and we're back. And this is the very next slide. So here, this image, rather complicated sequence of lines. What it denotes here is the transcriptional relationships between various segments on chromosome 22. Okay, so this is where scientists have gone in and pinned down where specific tissue proteins in the testes and prostate originate from on chromosome 22. Okay, you see something interesting here. You see that the beginning of one transcriptional sentence is actually linked to a very distant part of the chromosome. You notice that all these connecting lines are specifically the sequence pattern of the exons. So you see that it starts maybe on one part and then skips to the middle, then maybe skips again to a third place. Not only that, but you also notice, so the colors, what they're denoting is actually plus and minus strand locations. So DNA is a double helix. It has two sides, right? And what this pattern indicates is that you can read it in one direction, or you can read it in the other direction. So that's something that's pretty interesting. It's not a static code. What we also see is that not only are they completely jumbled up in the location on the chromosome, but they also form related networks. So there's areas where where these related proteins are transcribed, which have sort of common regions, which suggest that they are participating in some sort of regulation at the level of the chromosome. Now, what this basically means, without going into all the specific details, is that the information on the chromosomes is very fragmented, so it hasn't been defragged, right?
I was thinking of the computer metaphor as you were talking about it.
In this case, I'm not fond of mechanistic,
Sure.
mechanical biology, but it makes things easier to think about in some ways. So, in some cases, definitely. Now, in this case, it sort of raises the question of what is the purpose of this? Why, nature is thought to be parsimonious, right? Things that are unnecessarily complex or energetically expensive have to have some sort of justification in the long term, right? In this case, the question arises, why this complexity? We'll try to answer maybe some speculative theories as to why this is happening. But the classic idea of a gene really has to be questioned. Is what this sequence of slides is really supposed to get at, because genes as such don't really exist. More precisely, the term denotes sort of an abstraction. Does that make sense? It's a complex functional unit that doesn't really have, or very rarely corresponds to a single stretch of DNA or a particular, delineated physical, sequence. It includes regulatory sequences. It includes, variable transcription, where different exons from different genes can be spliced together to form the end functional protein.
Oh, I see. So it's not, part of your point is that it's not just, it's often related to an instruction manual or almost like a scroll that you just read down the list, right? And
yes,
that's,
it's like a separate component. It's like Tinker Toys or Legos to an extent.
yeah. That's what you're getting at here, is that they take these pieces and they put them together in different ways, which is
Yes,
very
that's not
different.
clear from this diagram, but yes, there's something called, there's a variable, splicing of these exon segments, so that you can read, to make one particular mRNA for a particular protein, you take exon here, here, and here, right? Jumping around this diagram. But to make a different one, you combine two, maybe the first two, and then a completely different third one, theoretically.
Right.
Like that happens a lot.
So, I mean, that alone would really mess with any, it seems to me anyway, as a novice, that it would really mess with any deterministic idea, because DNA is usually, in my head, the way I think of it before was, okay, it's supposed to be like this instruction code, like code, that just goes down the list one thing after another. But what you're saying is that it's able to, I mean, that itself is almost, it's like, how would it know to grab this piece or that piece if it doesn't have code to metacode to tell it to do that? Am I going off kilter there?
No, that is absolutely correct. That is,
Okay.
your intuition is getting ahead of us, but that's exactly where I'm going with all this.
Okay.
So that there is the fact that it's not a one-to-one correspondence, right?
Right.
As many people imagine, does require a kind of meta information that's stored somewhere else. In the soma, in other parts of this, and other, as we said, maybe in the 98% of the genome that's not coding for protein, we don't know. There's a major gap in this because of, again, the philosophical damage that has been done to biology to a large extent, but also because it's just really complicated. Who talks about it? Vervaeke talks about combinatorial explosion, right?
Yeah.
We're getting to the point where we're digging into these systems of biology and in science, even in physics, where things are just too complicated to really explain in an intuitive human way. I mean, maybe some supercomputer would be able to process part of this mystery, but it still wouldn't be interpretable. In any case, it's this is where we're at. I mean, this isn't a particularly, this isn't from some particularly controversial paper. This is from pretty basic molecular genetics. Yeah, so these scattered pieces are being put together by the cell in a way that's very very complicated. Even the classic idea of a reading frame that many of us learned in biology 101, the idea of a fixed reliable start and stop codon, applies to maybe at most 10 to 15% of eukaryotic coded regions.
Mm-hmm.
So really a tiny sliver of the pie. And there are all these, as you mentioned, there's these, it works like Tinker Toys. They're called chimeric transcripts, wherein the cell takes a little snippet of code here, a snippet of code here, a snippet of code here, puts it together and makes the mRNA that codes a particular protein.
So that's what's crazy to me, is that it's literally, it's meta coding. It's taking bits of code and it's creating new code for itself to tell itself what to do, is what it sounds like. I don't want to get ahead of, jump the gun or anything, because I'm obviously excited and I can get a little new agey about things, so I'm trying to restrain myself. But that's certainly what it's sounding like.
And that's just at this, and that is just at this level of the system, right? So this is just at the level of transcribing the DNA to mRNA.
Right.
If you go beyond, if you zoom out a little bit and talk about actually what happens to the mRNA, now, then that can also be trans-spliced. So then you take these mRNAs that are the product of this chimeric transcripts, and then they can be rearranged.
Right.
So there's a whole other level to this, where and it does happen. So, and not only that, but there are places where information on one chromosome, an mRNA is taken from one chromosome and then finished off with a sequence of information for another chromosome, right? And there is a communication between the unwound chromosomes in the nucleus, and picking of information between them. So I mean, this diagram is limited because there are segments that are not just limited to chromosome 22,
right? That go off to other chromosomes.
Right? So this network is even more complicated. And then there's other exotic methods of DNA transcription, which kind of differ from the textbook model, which we don't even have really time to go into.
Yeah, sure. I get the idea, though.
There's a lot of things going on. Now, kind of getting back to the point of all this, which is to talk about non-random mutations, right? So now let's just assume that all these complicated biological mechanisms are, forget about all those. Let's just talk about sort of the biochemistry of the double helix system to begin with. Now, let's say that there's no cell acting on this. We will just consider the nature of the chemicals that make up DNA. So what are they? Like adenine, guanine, thymine, and cytosine are the bases, the nitrogenous bases that make up the sort of the links that connect to each other across the double helix. There are 4 of them, right? As I mentioned, A, G, T, and C. And A connects to T and G connects to C. Now, adenine and guanine are purines, and thymine and cytosine are pyrimidines. That's just what they're called, the type of molecule that they are. In mRNA, the thymine is replaced by another slightly different molecule called uracil, but it basically is the same thing. Now, if to code for a particular amino acid, so to go from the DNA, mRNA, protein, the way it's translated, the way the DNA or the mRNA is translated, is that a particular sequence of 3 nucleotides, so A, G, C, or whatever, a particular sequence maps onto a particular amino acid. Now, there are 21 amino acids, but there are more than 21 possible 3 nucleotide combinations. Now each of the 3 nucleotides is called a triplet code or triplet codon. Now, what this means is that there is a particular correspondence between this sort of language and how it's translated into the amino acids, right? Now, keep that in mind. If we're going to talk about sort of the modern synthesis, right, and the shifting allele frequencies model, this kind of implies that absolutely novel changes do not occur, right? All variation is built into the initial gene pool, and evolutionary adaptation occurs via these changing allele frequencies, right?
There's a perfect continuity right from the bacteria up to the human being.
Yes, in a sense, right? Right. It's just all a response to evolutionary pressure. Now, the population view of evolution really says nothing about how an individual's novel phenotype arises and spreads in the population. That's really one of the troubles with this theory, and how attempts to link it to molecular genetics has actually stifled really the explosive, theories that molecular genetics should lead us to.
Interesting.
But while mutations are sort of viewed as necessary as a source of variation, the actual pressure that causes the adaptations, right, it's thought to be all external. So the mutations themselves are claimed to be incapable of influencing the evolutionary direction. You see what I'm saying? So the mutations are a completely separate thing from the adaptive process.
Right.
And it's mocked in the literature by the likes of Dawkins, the idea of a lucky mutant model. It's considered sort of a heresy in the evolutionary literature.
Just to make sure we're clear, the heresy here is the idea that the environment would somehow cause the mutations to happen more often or cause a mutation to, is that quite what you're saying, or?
Partly that is, admitted, but there's an idea that the mutations themselves don't influence the direction of change in any way, right?
Okay. Mutations
are not themselves biased in any way that would be evident from the evolutionary record. So the evolution is purely a result
I see.
of natural selection. There is no unnatural there is no natural mutational selection within the organism.
Right. The idea being, like you said, the idea of the lucky mutation, you're not supposed to get a mutation that, oh, we need legs now. Oh, here's a leg mutation, or
all organisms develop legs. They're just biased to develop legs.
Right. I see what you're saying.
Legs are a very distant phenotype, but on the nitty-gritty point, like, is there some kind of evolutionary bias just because of the way that mutations occur? Right?
I see what you're saying. Right. So in that case, the point being that it needs to just be this soup, and it could theoretically become anything. It could become any creature depending on the evolutionary pressures. Yeah.
The soup itself has no particular bias.
Yeah. The anathema to them is the notion that that the human form is somehow a perfection or something like this, that things will tend to evolve into. They don't want that.
That's taking it
pretty far, obviously.
But what we're discussing is even like something very like at the basic genetic level. And now here's the problem with that assumption, right? Which is in the literature, which is crazy. This is what these evolutionary biologists used to talk about. Now, change is a two-step process, right? And what I mean by that is a bias in what kind of mutations occur before they're displayed to the environment for selection. That is to say, before they become the phenotype, right? First, the mutation has to occur, and then there has to be a phenotypic change that's affected, that's able to interact with the environment to cause the natural selection, right? Now, so if there's a bias, right, to these kinds of mutations before they are displayed to the environment for selection, that means there would be a bias in the direction of evolution itself.
Right.
And there's really no reason, except this kind of quasi-religious dogmatism, to suppose a priori that mutations cannot be restricted or localized or otherwise controlled in time and genomic space. Right? Does that make sense?
I think so. Right. There's no evidence to say that there's no bias. It's an assumption that crept in, a preference that crept in.
And so here I want to discuss the very sort of the most baseline kind of bias that can occur. So due to the very nature of the triple codon amino acid mapping. So not all amino acids have an equal chance of being substituted for one another. Again, because of just the way the language of the code. I have to just, you would have to look at it like a table of how it translates, but you have to take my word for it.
Sure.
Now, assuming
I trust you.
Yeah, sure. I mean, the paper is here on the. Oh
yes, you're right.
Cited on the slide, so listeners and readers are free to look. This is by, interestingly, of course, not by biologists. This is by computational physicists and statisticians. So now it's pretty obvious if you think about it, but not all of these mutational paths are identical in probability. Some transitions of a particular codon triplet code to amino acid sequence are just more probable. They have more paths to occur because of the way the language, the translation of amino acid codon to triplet codon to amino acid is, right? It's just built into the very language of this system. It's pretty obvious when pointed out, actually. So if you look at figure C in the slide, that graph kind of shows probabilities in different side circles for transitions of a particular amino acid to a particular other amino acid, just based on the how many ways you can how many different combinations of triplet codes can be switched into another. It's a purely statistical.
I'm sure if I looked at, the way it all breaks down exactly, it would be like, oh, but like I get the general concept of what you're
the general concept is what's important. It's not about the numbers.
Yeah, it's the logic of the system that if you can only do so many things, there's rules here, and those rules lead to tend in a certain direction.
Exactly. So that's already built in. Now, the question that arises naturally, I think, is this code then just random in the way that it arose? Or do the triplets that represent certain amino acids make sense for some reason? That is, chemically similar amino acids are, for example, more likely to substitute for one another in this mutational framework. So there's like a conservative bias, for instance.
Right.
Is that the case? I don't know. I don't remember, honestly, if this is something that they discuss in the paper. I think it's the issue they raised, but I don't think they're able to actually answer that question. Furthermore, if you look beyond the actual code itself, and you now begin to think about the chemistry of it, there's also another layer of issue. So if you don't even think about, going beyond the actual, just the pure statistical probability of the language translations between the triple codon and the amino acid. And if you look at the actual chemistry of the way that mutations occur, it turns out that there is a bias. So there's something called transition and transversion, meaning is the nucleotide, is the purine switching to another purine, for example, the T to C, or is it transversion? Is the purine changing to a pyrimidine, meaning like the T changing to a G? Now, again, this is all technical stuff, but there is a difference in the likelihood that these occur. We know this from, decades of genetic studies, in vitro, in vivo, that there's definitely a bias. So purine to pyrimidine transversions are more likely than A becoming G or C becoming T. It's more likely that A becomes C or G becomes T. Furthermore, there are actually areas of the genome which are called C-poly-G sites, CPG sites, that are kind of long stretches of C's and G's, cytosine, guanine. And they have an effect. It's called the CPG effect. And that results in a 30 times increased transition and 5 times increased transversion frequency in areas that just say CG, 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 specifically in primates. In other mammals, those rates are half of what I quoted. So that's kind of interesting. We know it's there. We know this bias exists. We don't know what it really means. But in this paper, the interesting thing is that they modeled basically the resulting pattern of amino acid substitutions, weighing like, all synonymous codons equally. So weighing the, the probability of the same 3 letters making a particular amino acid equally. He gave an interesting pattern with all these biases that they were able to calculate. And that gives you B in the figure, which is an even more asymmetric distribution of particular probabilities of transitions of amino acids than we would just assume, all things being equal from the language of the code. The resulting, interestingly, the resulting pattern then, this B pattern, explains, apparently, in the study, 45% of the variation in chimp to human individual amino acid changes. That's kind of interesting. So so if this paper is correct, right, in this thesis, they've basically demonstrated that almost half of the specific amino acid substitutions, again, we're talking about this tiny sliver, the 1.5% protein coding part of the genome, 45% of those amino acid changes that have occurred from a chimpanzee to a human are explained by this model.
Okay, so what this is, just to make sure I'm clear, so what this is saying, if the paper is correct, is that it would mean there is a bias that would explain the evolution from chimp to human, essentially?
Yes, it means that the mutational bias accounts for a huge chunk of the phenotypic change that we see in evolution. And this throws out, well, I mean, this doesn't just contradict the modern synthesis. This is actually also kind of anti-Darwinian.
Right.
Because it actually goes against the idea of...
Because it would mean it's not natural selection, at least purely. Natural selection alone. There is internally in the genome structure itself.
I mean, you can bend over backwards and sort of say that there is some sort of natural selection occurring through the processes of blah, blah, blah. But yeah, you know what I'm saying. I mean,
you
can twist the theory until it becomes meaningless. But at this point, this is clear evidence, I think, that mutational effects are not insignificant.
Yeah. What's interesting, just as a side note, I'm bringing in my novice knowledge of physics, but sorry, I shouldn't point at the screen. The C diagram, where it's more evenly distributed, versus the B diagram, where you have the pockets of inequality. It reminds me, I can't remember what it's called, but it's that photo of the background radiation of the universe, which is important because it shows that there are somehow inequalities in the, like, from the Big Bang or something like this. And the reason that's important is if there weren't those inequalities, then it would be impossible to explain how stars would have formed, because the gravity would have just been equally distributed. So I know I'm drawing it's odd for a lot of people, perhaps, that I'm drawing connections there, but it's the important thing is that the inequalities is what allows for the gathering of the material together. Gravity is obviously not the same thing as natural selection or evolution, but I just thought it was it helps me understand it better.
That's interesting. Yeah, there is certainly this heterogeneity, this uneven distribution, which kind of begins to, it's kind of the hook into which interesting observations demands an explanation.
Yeah, so that Dawkins approach to it, or the more orthodox approach, where it needs to all be even split, logically does not make sense, or isn't how it generally works. So you wouldn't be able to explain. It's not a perfect comparison, but it works much better when you have these biases, because it just makes it, okay, there's no way this wouldn't have happened eventually.
That's the weird thing, right? I mean, if you look at these, there's an inherent mutational bias. Now, Of course, most of the actual genomic differences between humans and chimps aren't in the individual amino acid sequences of that 1.5% of the genome. It's in these large structural ALU elements. We'll get to those.
That's interesting.
But it's really more to do with, obviously, the 98% of the genome that doesn't code for protein.
Right.
That's
the non-phenotypic, you could say, I suppose.
Yeah, I mean, not clearly phenotypic, we should say.
Right, yeah.
But yeah, that's actually where most of the differences between humans and chimps are. But nevertheless, I mean, if we're focusing on that narrow strip, I mean, this is still a very interesting work, I think. Bottom line is, yeah, so these built-in features, right, ignoring any kind of biological regulation of the mutation processes. This is just, this is just notable. This is just the language of the codes plus what we know from observation of how mutations happen biochemically. So ignoring any kind of biological regulation on top of it, it shows that evolution is not a random walk with natural selection being the driver, or at least the main driver, or the only driver. And there are inherent bias in the kinds of changes that occur. So if it's predictable to an extent, or maybe modelable and mutation driven, it means that natural selection comes later. It's a second step to what's going on. The idea of the supremacy of natural selection then should be brought into question, right? I mean, it's really blatantly obvious to a lot of people who are working in this part of the sciences, in molecular biology, bioengineering, biophysicists. But the general public continues to kind of be fed these sophistries by the evolutionary biologist establishment. But let's move on to the next bit. Now, we talked about the very kind of basic, we'll talk about that guy in a little bit, the very basic level mutational biases, but there's also actually inherent trends in the way mutations occur on all scales. So not just at the levels of individual nucleotides, but there are major unexplained variations in the mutation rates in different chromosomes, large scale, different tracts and regions of chromosomes, sort of medium scale, and on the small scale, on various specific runs and sequence sites. So, I mean, this is all very technical, and I would urge people who are interested to look at the papers. But just a brief overview of what I mean by this. So first, we'll start with the small scale. So the rate of simultaneous doublet and triplet codon changes, right? We talked about how one change can occur, but when two adjacent nucleotides change simultaneously, or all three change simultaneously, that would be like a doublet or triplet simultaneous change. Statistically, that happens far above the expected combined independent rate. Okay. Although it's still 2 to 3 orders of magnitude less than that of an individual nucleotide, it still happens much more frequently than expected. Okay, it's interesting, but again, not a big thing. Second, let's go back to that, what I mentioned, the CPG phenomenon, right? Where CPG sites mutate frequently, okay, where it says CG, 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 CG. These sites actually mutate a lot, and actually estimated to be responsible for 1/4 of all human point mutations, interestingly enough. And these sites are actually, because they mutate so frequently, they're less abundant. And actually, what the cell will do is it will actually protect those areas by methylating, methylation being, in this case, I'm talking about different kinds of methylation. So there's methylation of histones, which are the structural proteins of DNA, and that affects it. But there's also direct modification of the cytosine residues. They're actually methylated by the cell, the actual nucleotide, and that actually makes them more likely to mutate and turn into thiamine.
I see.
Very interesting.
Yeah.
And in fact, it can also be done enzymatically. So they can actually mutate these residues via cytosine deaminase. So there's an enzyme that specifically goes in and actually changes the C to a T. The cell has an enzyme that tags certain nucleotides and uses another enzyme to then mutate them. So right there, you have the cell's ability to mutate its DNA.
Yeah, right there you have the backwards. Yeah.
Enzymatically, right? Protein is modifying the DNA. Exactly. So there you go. Now this increased mutation rate at these sites also bleeds into adjacent sequences. So it's sort of, if there's a CG like sequence, the areas around it also tend to mutate more. There are other types of sequences that have above average, like 3 to 4 times. When I say above average, I mean 3 to 4 times higher mutation rates. ATTG, AG, ACAA. They're variable context effects where specific nucleotides in the context of other nucleotides have higher or lower rates of mutation. This is all very well documented. Small nucleotide polymorphisms, SNPs. These substitutions tend to occur in parts of similar sequence sites across different species, which sort of implies that there's something going on sort of across the evolutionary lineage. There's a lot of weird stuff. And now on the larger scale, I mean, in the interest of time, I'm just going to skip to some of the chromosomal stuff. But I mean, I can go on. Basically, the point of this is that there are, at all levels, at all scales, there are changes, there are biases, there are ways in which the mutation rate is non-homogeneous. But what I think is kind of
built into the system, into the way that they combine and stuff, and also
and there's no way, and I mean, as I mentioned, there are specific ways that the cell works with this. So this is random. For example, going on to like the overall big structure, we know that that the DNA that's transcribed more, so the DNA that's used more, actually, to code for proteins, has a much lower mutation rate than non-transcribed DNA.
Oh.
Right. So the junky DNA likes to get funky. That's one way of thinking about it. But the one that's used and is coded for like specific structural proteins, that tends to have much lower mutation rates. So
it's like
there's a functional.
They remember.
Like the cell knows. Yeah, it's like the cell knows what's going on. Surprise, surprise. Now, the opposite is, well, not really. Let me put it this way: in germ cells, so in the ova and in the sperm, or in the progenitor cells of those, there are actually subtle differences in the kinds of mutations that occur. Maybe not the overall rate, but there's a completely different look to those kinds of mutations, where they occur, and how that stuff functions. It's kind of weird, but finally, there are large-scale variations at the level of the chromosome, which is what I wanted to discuss. So the overall effect is subtle, but statistically robust, such that, for example, the human Y chromosome, for males, has a 50% higher mutation rate than the non-sex determining chromosomes. And those non-sex determining chromosomes, the autosomes, have a 30% higher mutation rate than the X. So the X chromosome is by far a much more conserved chromosome in human beings and primates. The situation is completely different in even related mammalian species like rodents. I mean, their X has much higher mutation rates. Interesting. It's weird. I don't know, maybe there supports some sort of observation of why males are kind of evolutionarily experimental or expendable.
Yeah, I had heard that notion, which I thought was very interesting. And I could easily draw sort of broad comparisons with, I don't know if you, I think you watched my video on theorizing about sex differences from sort of this pseudo-Freudian or quasi-Freudian standpoint.
Oh no, I would like to. That's very interesting. But yeah, this is
anyway.
This is a correspondence that, you know, I'm familiar with that kind of idea, of course, in the evolutionary literature. But I thought that this genetic mutation rate bias seems to correlate, which I thought was interesting.
Well, I do know, and you probably know as well, that sort of classic notion that's been brought up a number of times, that for the statistical variation for women across a large population is like this, whereas you get variation of men it's just simply wider. So you're more likely to get both someone, to use IQ for all its controversy, you're more likely to get a man who is either really, really high in IQ or really, really low in IQ, whereas it's more conservative for women, the bounds and limits there, statistically speaking. And that would link up perfectly with this. Yeah, it's interesting.
And what I want to discuss here is a couple interesting observations, what are called hot genes. So there's this observation found by DARPA-funded researchers that it seems that mutation rates, if not just all these structural factors, but there seems to be that if you measure sort of neutral substitution rates, sort of the rate of silent mutations that in the triplet code that do not change the amino acid sequence, they usually occur in specific areas of mRNA or of the DNA that eventually gets into the mRNA in specific areas. And they're kind of, it depends on the type of gene. So if you split the genes based on genes that are kind of dealing with environmental interactions like smelling, like immune response, immune interaction, various kinds of secretions, venoms, versus kind of regulatory housekeeping genes, kind of ones that are involved in internal homeostasis, there seems to be a big difference in these kinds of silent mutation rates. It's just they're easier to measure is why they're doing these silent mutation rates. But what's real crazy is that when you look at mutation hotspots, there's the cone snail, probably heard of that. It's a small venomous snail that it's predatory, and it's got crazy venoms, very interesting from sort of some medical applications. But they've identified basically that these they have these areas in the gene that codes for the venom. That is, there are areas that are extremely conserved between species, just that one particular area, and then others that are extremely variable. And it seems to be like there's sort of an accelerated evolution that they see, which is in natural selection in this case is irrelevant because they're looking again at what I mentioned are synonymous substitution, meaning areas that changes that are phenotypically silent.
Right.
Those are highly elevated, and they're elevated in a very particular segment of that gene that involves kind of like the.
Oh no. Ilya, whenever you hear this, you have frozen.
Is really attacking.
Hey, Ilya, you froze for about 5-7 seconds.
Oh, whoops, that's a while.
Yeah, so right about elevation, you were talking about they're elevated.
Elevated. Oh, right. So where the elevated rates of mutation? Right.
Yes, I believe so.
So in this cone snail, different species, they have a lot of different species, and their venom, the gene that codes for the venom, seems to have an area that is hypermutable,
Right.
that has a lot of mutations and changes. And again, this is all measuring synonymous substitutions. So these are
Right.
changes in the codons that don't result in change in the amino acid. Because if it did, then this would be another, it kind of defeats the purpose of looking for this kind of mutation rate, so to speak. It has to be phenotypically silent. So if you look at these cone snails, they have areas of this venom gene which are highly conserved, sort of the structure, and then that one particular area of the gene seems to be highly mutable. So there's not just differences between different genes and the function that they carry out in the cell. There's not just differences in the mutation rates between those, but there are mutation differences even within a particular genetic sequence for a particular protein.
Mm-hmm.
Which is quite amazing, was quite unexpected, but makes entirely all the sense in the world if we think that the organism knows what's going on, knows what parts of its DNA do what,
right,
and is able to regulate that.
Right.
So if we take that as a given, then these things aren't, surprises, isolated surprises. This is what we expect to see.
Right.
And that would really throw a wrench into our biological models that are working, that are, people have been doing for decades. I urge people to look at the literature cited here if they want more details, but I'm trying to give an accurate summary as best I can. Now, this is where we kind of blow things up further, and we get to, so far we've been mostly talking about that tiny sliver of the genetic code, right? That tiny 1.5% that codes for actual proteins. So here I'd like to get into finally what 2/3 of our genome is composed of, which are mobile elements. Now,
mobile
I think,
elements.
as I mentioned, mobile genetic elements for proteins, but quite simply, these are sequences that move around the genome. Okay, so right now in your cells, particularly in certain organs, including your brain, and if you're a man, your testes.
Mm-hmm.
There
particularly
are cells,
in the brain and testes, you said.
Particularly in the brain and testes, and we'll get into that. But there are cells which are having their DNA modified by these mobile genetic elements. They're moving around, and they're changing, they're re-engineering your DNA. Now, they come in different types, many different types. There are ones that copy themselves and reinsert themselves somewhere else. There are some that cut themselves out and reinsert themselves somewhere else. There are some that cut themselves out and then recombine, meaning sort of a different process of insertion where kind of certain areas are canceled out. Some of them encode their own enzymes that allow them to do this sort of transposase or retrotransposase, meaning that they convert themselves into mRNA, then that mRNA is converted back into DNA and reinserts itself. So some do that, some don't do that. Some rely on the enzymes, the transposase or retrotransposase of other mobile elements. Okay. They come in different shapes. Some are linear, some are circular, some fold in different ways when they cut themselves out. Some duplicate themselves before reinserting. Some are non-duplicative.
So the DNA, and pardon me if
sorry,
I'm
they're
jumping.
duplicative in regards to adjacent sequences. My bad. They don't duplicate.
Sure.
Yeah, but they do duplicate themselves sometimes. They cut themselves out or they copy. Yeah.
The bottom line here being, and excuse me if I'm jumping ahead, but the book of the DNA is rewriting itself. It's the DNA which is doing this.
Yes, yeah, exactly.
The DNA isn't just passive. It's like a living thing, if you will.
In fact, 2/3 of it are this. 2/3 of human DNA. In other animals, it's different numbers. But 2/3 of our DNA is this kind of mutable, like roiling, twisting, coiling thing.
That's crazy. How was this not seen before? I guess because it always dealt with dead DNA. Okay.
This Nobel Prize winning discovery by one Barbara McClintock. This is known. There's a whole database of these. You can go on to, I don't know if it still exists, gydb.org. It's a whole university database of mobile genetic elements. New ones are constantly being discovered. This was supposed to be probably, well, I mean, part of this is it's a very interesting story of her personally, but so these were originally called transposable elements or jumping genes. They were discovered by Barbara McClintock back in, I should know this for the presentation, but it's a while ago, in like the late 60s, I want to say. And they were speculated initially to exclusively be just a curiosity in corn. So she discovered this in maize, and everyone was like, oh, okay, whatever, right?
No one cares about corn.
No one cares about corn, apparently. Which is strange because it's massive, biomass of farming, destroying the Midwest, prairies, but different point. It was speculated really in her Nobel. Let me quote you her Nobel Prize speech, and this gets to the point of why this didn't take off. So she initially speculated extensively on their role in gene regulation. She believed that this was her major contribution, really predating kind of the operon theory from prokaryotes. She believed that this was like kind of the major genetic regulatory method. They were then discovered in bacteria 20 years later, right? And in fact, are really largely responsible for carrying some of the drug resistance genes and other interesting things. So she won the Nobel Prize in 1983, years later. And in her lecture, she said, quote, "There must be numerous homeostatic adjustments required of cells. The sensing devices and the signals that initiate these adjustments are beyond their present ability to fathom. A goal for the future would be to determine the extent of knowledge the cell has of itself and how it utilizes this knowledge in a, quote, thoughtful manner when challenged. In the future, attention undoubtedly will be centered on the genome, with greater appreciation of its significance as a highly sensitive organ of the cell that monitors genomic activities and corrects common errors, senses unusual and unexpected events, and responds to them, often by restructuring the genome."
Well, that throws a lot of things out the window.
This
if
was
the DNA
Nobel
isn't,
Prize lecture that she gave in 1983.
That's insane. When was the selfish gene written? That was like the 90s, wasn't it?
Right. So
Maybe it was
this
the
is
70s. But the
uh,
point, yeah.
yeah.
Uh, I don't want to. I don't want to get. I think it was in the 70s, so I'll forgive Dawkins, I suppose. But but I don't want to be mean. But the okay, yeah. So
He said this in 1983. This is exactly what I'm saying now.
yeah, yeah,
Right? Pretty much. So this is like what 40, what, 20, 40 years later.
yeah, yeah.
Yeah.
Yeah,
So
because
invariably,
it throws a ton of stuff out the window. I mean, the idea that just that idea that the DNA can be considered or should be considered as alive in its own right, as you know, in the sense that it's moving and changing itself. And
mhm.
her feeling clearly was that it's a sensitive organ. It should be considered more of like an organ, the chromosomes here that
Yeah,
change
yeah, it's a
themselves
cellular
in
organ.
response. And that
Exactly.
notion completely throws out the normal notions of evolution, because it makes it so that that stuff at the bottom that everything is being created out of has a certain, and I don't know what else to call it, it has an intelligence to it.
Yes, exactly. Now she clearly was ahead of her time. But to avoid sort of upsetting the rotten apple cart of this modern synthesis, um, her discovery was essentially lumped in with the selfish gene hypothesis, which is how
Oh,
many people know of these, transposable elements. It erroneously implies that they act randomly
oh, I see.
or that they act selfishly. That's not true in the least. Okay.
Uh-huh.
This shoehorning of the sort of uncomfortable fact of this massive genetic plasticity into the dogma of random, independent genomic behavior, uh, I mean, set the field back decades, I think. It was really all because of, again, the fear of agency in living systems, as you allude to.
So
It
it
would be
was
unmaterialistic.
Dawkins' fault.
Yeah,
Or the
yeah,
selfish.
I do think so.
Okay.
Yeah, science forbid this kind of thinking. Um, the truth is, the movement of these mobile genetic elements is not random. Okay,
Okay.
most are targeted to specific sequences. They tend to insert themselves in areas where they don't disturb function too much, or at least in any critical way, a critical, obvious way. And they are regulated in the sense that they're only really allowed to function under certain conditions. So they are very active during early embryogenesis, the brain and the testes, as I mentioned.
The brain is
And
very interesting that they should be
it is
active there.
interesting. It is very interesting. And during cellular stress.
Mm-hmm.
Very interesting. Now, the embryo, the brain, the testes, that's very interesting. Stress, you could argue that that's a sort of de-repression. Maybe it's, I mean, you could shoehorn that into the selfish gene hypothesis, but not the other stuff. And again, I would still be ignoring big chunks of the evidence. They contribute widely to genetic regulation. So a lot of areas of the genome that have promoters, enhancers, splice sites, and that kind of areas that have amplification of certain exons. These areas are linked to these mobile genetic elements. So these are areas where they kind of insert themselves. In fact, there's a very interesting example. So in yeast, which have there's several kinds of these transposable elements in yeast, which target specifically, they only go in areas that are upstream of start codons, sort of avoid messing up the reading frame. Or they only go into untranscribed regions to avoid, again, disrupting transcribed areas.
If it was random,
Yeah,
you would have them doing that all the time.
exactly. But when the cell is stressed, some of them do that, but only when the cell is
Oh,
stressed. So
Makes
much for
sense
yeah,
if there's this, if we're dealing with almost kind of an intelligence and almost anthropomorphic kind of thing where you can, because that makes sense intuitively, where it's like, oh, it's stressed out, it's messing up, if you will. Maybe that's jumping too far, but
could be, or this could be a way of the cell to try to get out of a stressful situation,
or that.
kind of adapt itself, accelerate its sort of its ability to change. In any case, the fact that they're, that they have this kind of behavior, if you will, really speaks less to their selfishness and maybe their downright considerateness as genetic sequences. So the same is true in other eukaryotes. They behave like helpful genes. They seem to be involved in all these networks. Makes you wonder if Dawkins was maybe projecting his own character onto the onto the subject of his work.
I wasn't going to say anything.
But getting back to, like we mentioned in variable transcripts, the same thing here. These work like chunks of Tinker Toys or computer code that may represent like a single functional motif that can spread itself in various parts of the genome. So for example, in higher primates, we have this increased frequency of what's called ALU, A-L-U, retrotransposon, which is a special kind of mobile genetic element, which transcribes itself into mRNA and then puts itself back as DNA somewhere else. As you go up the primate lineage, the number of these increases. So humans have a lot, but it actually relies on a retrotransposase from another kind of mobile genetic element called line element, L-I-N-E. So what's interesting is that these famous ALU retrotransposons have been observed in the endpoints of 30% of segmental duplications, low copy repeats. What this means is these are structures that are involved in sort of rare recombination sites that really only increase in humans. So they're kind of rare in mammalian lineages, but they seem to happen more and more frequently in humans. So there's this very odd, but very clear evidence that as we go up the evolutionary tree to humans, assuming that we're more advanced, I think we can reasonably say that we are, the number of these ALU retrotransposons associated with these other rare, with these rare genetic structures seems to go up. What does that mean? We don't know. But
it's an interesting correlation.
It's an interesting correlation, but they are associated, we do know that they're associated with sort of binding sites for nuclear scaffolding proteins. Suggests that there's a very high level of regulation on the actual shape of the untangled DNA. So they're actually providing kind of links throughout the structure where the nuclear scaffolding proteins attach. It's associated with certain classes of mobile genetic elements. So it may be creating like a standardized structure in the actual physical layout of the chromosomes. Very, very interesting stuff. Again, this is sort of a exploding kind of area of interest. Yeah. There's a lot of various, this is, cutting edge stuff. So we don't really know a lot, but there's a lot of weird observations. Very interesting. Yeah. So it makes sense that when a cell, for example, is stressed, including like nutritional deprivation, there's other things which that will increase these activity of these transposable elements. There are certain kinds of intracellular signaling molecules which will also increase their activity. Different toxins, including not specifically DNA damaging toxins, will actually induce this activity. Certain kinds of infections, and various kind of events that, in a broad sense, seem to disrupt certain epigenetic control systems. It's very, very interesting. And the association between these mobile genetic elements and sort of the evolutionary lineage really requires us to expand our understanding of what this all means, because we really don't have a good grip on it. There are great theories out there. There are people who are speculating, trying to put it all together. There's a guy at the University of Chicago, what's his name, Shapiro. People can look up his website and book, where he talks a lot about these in regards to his theories of sort of biological self-engineering. I mean, there's a lot of people talking about this stuff. So there's they're associated with telomeres. They're associated with centromeres. They're associated with basically large regulatory structural aspects of the chromosome. The VDJ recombinases. Those are the enzymes that allow immune cells to modify their own DNA to create antibodies that are variable and that attack different epitopes, right? So our immune cells change their DNA in order to create antibodies, right? I mean, that's how they create the antibodies. You have you have a different protein sequence, and you have a different genetic sequence. So they have methods of modifying their DNA to do that. And those structures and those mechanisms are associated with, again, mobile genetic elements. The idea of imprinting, so the idea that there is some sort of ability to pass on, sort of in a Lamarckian sense, inherited or experienced characteristics seem to be associated with some of this work, sort of stressors that are transferred to the germline. Pregnancy, so mammalian pregnancy and the actual creation of the placenta and syncytians and all this stuff would be impossible without the contribution of retrovirus-like mobile genetic elements that are involved in creating these structures of the placenta in mammals. Interestingly enough, different placental mammals have different mobile genetic elements associated with some of this stuff. It kind of varies by lineage. It's kind of weird.
Yeah.
So there's a lot of very important things that we rely on that are related to these mobile genetic elements. How are they regulated? Well, as I mentioned, they're not random, okay? And there seems to be a lot of control at the cellular level. So there are special kinds of interfering RNAs. They're called Pee-wee interacting RNAs. They're found really found in the mammalian testes, but seem to be active in other places too. But as we mentioned, the testes and the brain, the testes specifically are transcriptionally hyperactive, meaning a lot more DNA is transcribed into RNA than we know codes for protein. So we don't really understand what's going on there. Why? Is there some sort of meta level reprogramming that's going on? That's one reasonable hypothesis based on this view.
Would it be something along the, I mean, would it be at all related to the earlier idea of the male being more experimental, or,
maybe, but it also happens in the cells that become the ovum to a lesser degree, but it still happens. It may be that it's a matter of degree, probably. It's very possible that it's probably more pronounced in men. I don't know. I can't answer that for certain. I do know that it does happen in the ovum. I don't know what the differences are. I mean, plausible that it should be different. Now, these interfering RNAs seem to originate from repetitive regions that affect all kinds of DNA methylation, epigenetic regulation, RNA silencing. There are other pathways that regulate transposon expression, also RNA related. Classically, any kind of stress or genomic shock kind of de-represses their activity. And it seems to really it's really a de-repression. So there are different, obviously, ways of regulating. You can activate things in different ways. But in this case, the stressor seems to, the cell has various mechanisms to break and suppress these mobile genetic elements. So there's like a tonic pressure on them.
Right.
And when it's under stress, this tonic pressure is let off, and sort of
Right.
it's allowed to
Like
brew
lifting the lid off of a kettle.
Yeah, you can think of it that way.
Sorry.
Exactly. And different stressors result in different kinds of transposon activity. So it's not identical. So it's not non-specific stress response. It's a different kinds of stressors seem to cause different kinds of transposon activation. This is likely a very ancient way for cells to adapt to environmental pressures. I mean, life has a will, and this will is expressed in its very fundamental nature. I mean, this isn't just a matter of, as we mentioned before, dying and letting the pre-existing lucky mutant flourish.
Right.
So a sublethal stressor will induce adaptation at the genetic level. As I mentioned, there's various gene networks that seem to be, that are hypothesized to be related to a rise due to the help of a special class of large transposons called endogenous retrovirus elements. Things like placental signaling cascades, the P53 network, which is a way of sort of a network of cell proliferation important in cancer cells. These are related to these retrovirus elements, which compose about like 5% of the human genome. So, 2/3 of the human genome is this mobile genetic element, and 5% is this kind of specific type called endogenous retroviruses, or long tandem repeat retro elements, synonymous. Yeah, I'll skip over some of the technical aspects in the interest of time. But one interesting thing is that, let's see.
I will admit.
Yeah, I have a human brain specimen. So one thing I do want to mention is that there are areas of the human brain that seem to have active retrotransposon activity. So it's been well studied with transgenic mice and in vitro cultures and in actual human brain specimens. Now, the patterns appear highly variable by region and by individual, and are really hypothesized maybe to be related to certain neurologic disorders, including autism in some papers. But of course, what hasn't been linked to autism these days? In fruit flies, there is a sort of a class of neurons which are related to olfactory memory, which have been very active, observed to have extremely active mobile genetic elements. All this, the significance of all this, crazy genetic engineering is really a mystery. And some suggest that that poor regulation of these and insufficient suppression is related to certain genetic conditions, and they may contribute to a number of human diseases, but not clear. Really, it seems to be mostly restricted to neurons in the brain, rather than astrocytes and glia. So astrocytes and glia tend to have lower levels of this mobile genetic element activity.
The fact it's in neurons is extremely interesting to me. I'm going to have to look more into that.
Yeah, yeah. Specifically in neuron progenitor cells. So
okay.
It's early in the neurons.
Oh, okay, I see.
Differentiation. But these neuronal stem cells are active throughout life. So there may be something that's seemingly going on even in late adulthood with this. But it is very interesting that it should happen in the brain and in the testes. Again, there's a lot of, you can speculate, but there's not a lot of links to what we could speculate on. So I'll leave that up to you if you have any hypotheses. It's as valid as anyone else's.
I'll keep that in mind.
Yep. Now, yeah, I'm going to skip some of this technical stuff. Oh, here's a very interesting chart. I do want to go into this because it's a little bit funny. So there is something called zinc finger endonucleases, which are enzymes that chop up DNA. So it seems that if we look at different mammals, so this last chart, these dots represent different mammalian species. And on the, on one axis, we have the LTR retroelement number. So these are long tandem repeats. These are the endogenous retrovirus-like, mobile genetic elements. And then on the x-axis, we have zinc finger endonuclease number. So you see that there's a rather strong linear correlation between the number of these mobile genetic elements, the specific type of mobile genetic elements, and these zinc fingers which chop up the DNA. So it seems that the more of this, this roiling viral-like capacity that you have to your genome, the more of these regulatory enzymes you need. So humans are kind of in the middle top there. There's one animal. So that animal that's out there on the far top is a very interesting animal. It's the possum.
Okay.
So if you've ever wondered why possums are weird,
this might be a reason.
This may be a reason. They have they far exceed humans in the number of these long tandem repeat retro elements.
Well, that's interesting. All right.
Yeah. Yeah, moving on. It's another thing about possums. Their blood contains a universal antivenom that basically destroys and binds all proteinaceous venoms. This was discovered by I forget her name. She was an Indian researcher at Rice University, I think, and she even patented the protein sequence for this universal antivenom. Huh. Very interesting.
Yeah. Lucky possums.
Yeah. Now, here we can talk about a little bit, sort of maybe an extension of some of what we discussed. And it's sort of the question of where do novel genes arise, right? We discussed this as one of these paradoxes in this modern synthesis view of evolution, which is if new genes are modified old genes, how do completely novel, never previously encoded genes arise? Okay. Because it would take multiple steps for a gene to arise from non-coding material. Seems likely that it would require several steps. So how are these multiple changes selected in nature and propagated in the population, right? You can't just have one lucky mutant who gets one change and then happens to have, produce, and then one of those, inherited organisms also happens to get just the right mutation, and then so on and so forth. It's absurd.
Yeah.
There has to be a mechanism by which silent, genetic material is able to change and propagate in a population. And if they occur in some sort of protogene, doesn't affect the phenotype, what's the mechanism? We don't have a mechanism exclusively through natural selection. So natural selection cannot be the only thing that goes on. It just doesn't make sense. These are all uncomfortable questions, obviously, for defenders of the modern synthesis, but they basically deny the existence of de novo genes in various ways. They invoke genetic drift sort of as a mechanism for fixing undetectable traits in a population, or undetectable, phenotypically silent, I should say, traits. All this is equivalent to saying it just happens to work out. There's just
yeah.
Now, observations of evolutionary lineages suggest that genes do arise in multiple steps and spread in the population while apparently still unripe for the sort of prime time performance in the phenotype. So how exactly a particular configuration of protogenes becomes fixed, is the term, in a population without natural selection is not clear. So one theory suggests that these cryptic regulatory signals of some kind exist in all intergenic regions, all the introns and basically the 98.5% of DNA has some sort of regulatory sequences that kind of allow the organism to know what's going on there. So and this was a while ago, and I'm sure there are other theories, but I stumbled across, when I was researching this, again, this is from 5-6 years ago, something called the writing phenotype, which was by a researcher at sort of theoretical paper from Virginia Tech. But if you look up the writing phenotype, it kind of talks about this. Briefly, the theory states that genetic data encodes onto another chromosomal region, allowing a higher level of selection based on a sort of metagenomic encoding. So there's like a silent allele that describes what combination of alleles or non-transcribed DNA exists at another location. That's a very interesting idea. I'm not sure that the process of this is exclusively genetic. I think that what's more probable is that while this may occur to some extent, I think that's what's more probable is that some of this information is maintained epigenetically or in the protein and RNA mediums of information storage, so to speak. I think he was trying to get at this to kind of minimize the impact on the central dogma kind of idea. But right. I think we should just throw it out the window because, yeah, that's what I think. The brain and balls hypothesis, as I call it, or
that's yours. Okay, I wasn't sure.
You can also call it nuts and noggin, cabeza y cojones, or telencephalon and testes. I was obviously had. I think I wrote parts of these notes while drinking.
Oh, okay.
I'm pretty sure. Just a little bit like after dinner. Yeah, I'm pretty sure it was chartreuse too. But this hypothesis, you can name it in various ways, states that sort of novel genes tend to be expressed in novel tissues. Right. Not implausible, sort of at the cutting edge of adaptive pressure. So the male mammalian reproductive organs, and of course the newer parts of the mammalian brain, for example, are certainly at the cutting edge of evolutionary adaptation, competition. And so it's not unsurprising, right? I mean, it's not surprising, I should say. It's not surprising that this is where we see sort of more of this genetic activity, right? I can go in some of the observations. There's a particular number of particular papers that sort of try to trace out how genes arose. They kind of go through the mechanism, but they're obviously, I think they're kind of unambitious in their conclusions and how this affects sort of bigger theories in biology. But to quote Nietzsche briefly, on the slide here, the utility of an organ does not explain its origin. On the contrary, for most of the time during which a property is forming, it does not preserve the individual and is of no use to him, least of all in the struggle with external circumstances and enemies. So this guy, he was already clued into the fact that natural selection is not a complete explanation for the origin of species.
Yes. Yeah.
It's very interesting. So the thing about the writing phenotype, why I wanted to mention it as a hypothesis, is because it seems to actually explain the prevalence of sex biologically. So sex is actually the predominant reproductive strategy in all organisms. Even asexual organisms have sexual periods or occasions or capable sometimes. It's often assumed that sex evolved later and that asexual reproduction is sort of the early default. But we don't know. That may not be really true. Because sex, strictly speaking, is just genetic recombination and exchange. There's no definitive theory as to why it even exists actually and why it's so common and apparently necessary, because most of the existing asexual groups of any organism are pretty recent in evolutionary history, and they're likely, we don't know for sure, but they're likely short-lived. They're probably just quote unquote broken sexual organisms. The advantages of sex are not obvious actually from an evolutionary standpoint, because if you have a good combination of alleles, why would you risk breaking it up?
Right.
And then creating some other combination which may be inferior. And you can have plenty of genetic, well, we'll talk about how you can actually have genetic variation and genetic exchange without sex, called horizontal gene transfer. But that's the advantage of this hypothesis, this writing phenotype hypothesis, wherein, as I mentioned before, the overall genetic information is encoded into a sort of condensed meta genetic sequence where it kind of contains information about what's written everywhere else. The advantage of this writing phenotype is that it would, if it exists, if this writing phenotype is really a feature of genomic variation, it would sort of mitigate the loss of fitness in a new combination. And by this I mean basically the previous combination of alleles would sort of leave a coded archive on its germ cell genome, and that would maybe could be matched up against a sexy complementary archive on a partner's, on a partner that has a, allele archive that's compatible in a way.
Right.
This could allow for sort of abstracted second order selection for a better combination of genes rather than individual genes themselves. Does that make sense?
I think so. The driving point here being, again, it's as though because normally the theory of evolution, one of the things that bothers me about how it's explained or how it's presented is it's as though it's like, why would, and it's as though they're trying to explain how things degraded or entropied into more complicated forms down the line. Like, because it's like, why would anything ever stop being bacteria? Like, bacteria are amazingly successful. They just keep replicating a million times. But
many
if
ages which are doing fine.
yeah, exactly. Yeah, and so it's like, but if you put into it the notion of not only sexual reproduction but it makes it messes with that, where it's like, it's as though it's designed to improve itself. That's the thing that's crazy. But it's like, what constitutes improvement from a purely a purely a non-value, non-teleological evolutionary standpoint? Improvement doesn't mean anything. There's no such thing as improvement. It's just supposed to be happening, whatever. But this almost is as though the improvement is built into it.
Yeah, as though there were some sort of animating will to the whole process, right? It goes back to, yeah, what the organism's playing. It's not playing against, there is no, well, I would argue that in this case, we can argue there's no higher, there's no set of rules, right? There's
right.
no, like, the rules are what the telos of the
right.
It would make a lot of sense. That's a very good point. It's insightful commentary.
Thank you.
That's why we're doing this. The it also, the writing phenotype might also explain why we observe that so much DNA is actually transcribed in sperm cells. So not really because it's performing any specific, cellular function, but because it's being written into maybe other parts of the genome.
Okay.
That's, as we mentioned, sperm cells, and interestingly, cancer cells transcribe almost 50% of their genome. So maybe they're knocking out some of these, and it's interesting because if you actually go in and knock out pseudogenes, so you can do knockout models in organisms where you basically stop the transcription or translation or function at all, or just destroy a particular sequence. If you go in and into certain pseudogenes, genes that aren't active in other tissues, and you kind of knock them out, it does cripple sperm cells in certain of these models, which would seem to imply that this like over-transcribed mess is somehow critical for cellular function in sperm. But another way of interpreting this is to say that genes that are being actively written or are part of this read-write machinery are just critical to the gametes specifically. Anyway, it's an interesting idea. I just wanted to throw it out there and give this kind of obscure theoretician some. Someone is reading reading these works and trying to place it in a broader context. Yeah, these are.
Okay, we're back. Keep going. Had a brief interruption.
No problem. And so, as we mentioned, we talked about a little bit about sex and this writing phenotype. Going back to our main point about genetic modification, we've all heard of the CRISPR-Cas system, which is used. This is biotechnology breakthrough. Now, what this is actually a form of bacterial immune system. So it's called a type 2 CRISPR, which CRISPR stands for clustered regularly interspaced short palindromic repeats. It's a system that's used by bacteria and pretty much all archaea, so a big chunk of bacteria and most archaea, to provide immunological memory in case of viral invasions or any foreign DNA. So it works by incorporating short sequences of this exogenous DNA from the invading pathogen into specific loci on the host genome. Then it kind of, upon transcription, these sequences are processed into this kind of what's called pre-CRISPR RNAs, and then CRISPR RNAs, and they're matured into these kind of detectors of foreign DNA that can be targeted for degradation. So it's really kind of a bacterial immune system that's an acquired immunity that's recorded on the bacterial chromosome. And in fact, it's so widespread and so used that they're actually viruses that hijack this system for their own replication. But clearly, the bottom line that this is obvious evidence for genomic plasticity.
Mm-hmm.
And it actually corresponds to a similar method of immunity that's RNA based and epigenetic. It's not really genetic in plants and other eukaryotes that involves interfering and small interfering RNAs. So that's just major point of genetic modification. Another interesting thing going on the subject of bacteria is the fact that we don't just see the de-repression of mobile genetic elements in eukaryotes. An older and much bigger literature exists that shows the evidence for stress-induced mutation in bacteria. It's a very common response. It's quite variable, and a lot of evidence has accumulated that these mutations occur and in response to stress in bacteria, and may in fact be quite adaptive. So this research originally was pioneered by John Cairns and Barry Hall, and there's an excellent review article that I cite here. Although more recent articles maybe fill in some of the gaps, but if anyone's interested, it's called Mutation as a Stress Response and Regulation of Evolvability by Galhardo and Hastings and Susan Rosenberg. I think if you look up Susan Rosenberg, there's also a lot of, she's on a lot of other research involving this fact. Now, it's, I won't go into too many of the weeds, the technical aspects of this, but it occurs in really, and
you froze there for just a moment.
Mm-hmm.
What do they occur in?
It occurs again in a wide variety of bacteria, and it seems to be that different patterns of mutation occur in response to different stressors. So there's clearly some,
I see.
feedback mechanism between
Right.
what's being sent and the response. It's not a random or non-specific event. But we won't go into summarizing the literature. I mean, but this is kind of well known. Of course, there's also the epigenetic role of RNA. So there are quite a lot of types of RNA that exist. We are discovering new types all the time. There's the explosion in the types of papers. There's generally, when before a function is known, some of it's called non-coding RNA. There are huge swaths of what are called long non-coding RNA, different types in mammals. It serves all kinds of functions. But what I wanted to really discuss, what's very interesting, is there a particular class of single-celled free-swimming single-celled organisms, which actually have a independent heritable cache of sort of RNA that they pass on when they reproduce. So there's like a macronucleus, which is sort of active, transcribed, and then they have what's called a micronucleus, which is kind of their germline storage of information. And during reproduction, there's kind of like a RNA recombination and refinagling of the germline nucleus in response to adaptive somatic signals that the cell has received. So this is a clear, one very bright example of how RNA information flow facilitates this overall somatic information flow to the germline. Again, violating the central dogma, et cetera, et cetera. But I mean, we've found so many examples. It's just at this point that everybody, it's nonsensical to even talk about that as our goal. Instead, we should be forward-looking and seeing how these mechanisms imply sort of a new philosophy of life. I think that's the ultimate goal which we get to at the end of this presentation. But that's a very interesting little tidbit of biology, I think, this kind of micronucleus, macronucleus information catch in these organisms. Finally, we've all heard of epigenetics. This is essentially on top of the genome modifications, right? So it includes covalent chemical modifications of DNA. As I mentioned, methylation and deamination of cytosine, which really is not epigenetic. It's sort of direct genetic effect. But there's also modification of the chromatin structure of DNA. There's all the non-coding RNAs that kind of latch on and regulate it. There's all kinds of regulation of gene expression that relates to the actual structure of the chromatin, which is how DNA is folded. So besides as I mentioned, the methylation of cytosine itself, the actual DNA nucleotide, which serves as a regulatory signal, there is also broader kind of, evidence that this has clearly heritable functional effects in populations. So the big ones that are kind of famous are the sort of the Dutch famine studies. Showed that epigenetic effects were robust and detectable in humans. Now, there's a bunch of other animal models where this was done, but humans is proving something or demonstrating the possibility that it works in humans is a big deal. One of the first findings was that you lived longer if your grandfather starved around the time he hit puberty, but you had a shortened lifespan if your grandmother starved while she was in utero, so when her eggs were being developed. Anyway.
Which is similar to things that I believe Darwin toys with or mentions when in the early parts of the Origin of Species, when he's discussing changes with domesticated animals and changes that happen during the process of the creation of the gametes and stuff. It's a little, he's not up to date on stuff, obviously, but I remember being struck by the fact that he was talking about that kind of thing.
He was speculating on something that's been, seemingly demonstrated. Yeah.
Yeah.
Yeah. We have sort of the endocrine disruptor effects, which seem to echo into various generations. The effects of diethylstilbestrol exposure has been found to act that way. And there's even evidence for epigenetic effects of learning in mice, chickens. So which leads to some unsettling, social implications.
Yes, it does.
You basically, the experiment, to summarize, was that scientists grew these chickens under stressful conditions, sort of randomly fluctuating day-night rhythm, random access to food, and unpredictable access to perches. And so they were stressed and had really a decreased spatial learning task, like capacity. They became,
Interesting.
Became stupider.
Right. Yeah. Yeah.
Now,
Yeah.
and remarkably, the offspring of these birds, although they were grown in a normal environment, non-stress conditions, had the same learning disability, basically. Um, and they were also actually more competitive, aggressive, and they grew faster than non-stressed birds.
Mm-hmm.
That's interesting.
That's very
Um,
interesting.
yep, yep. There's also, sort of ideas of various kinds of, sort of the question of how relevant are these epigenetic changes when, uh, you know, this DNA or aspects of it, certain kinds of epigenetic changes are very dynamic in early development and the germ cells. So there's this idea that they're all erased, sort of at one point, but that's not really true. So this kind of, these graphs sort of show specific DNA methylation patterns of maternal and paternal DNA sequences during fertilization and very early development. So although they change a lot, we can't really say that they are irrelevant or that they entirely disappear. And again, this is measuring really one type of epigenetic marker. So this is an objection that I think I found in some of the discussions of epigenetics by conventionally minded evolutionary biologists, but they're really grasping at straws when they claim that, it's irrelevant. It's very interesting. It's always very disturbing when a scientist claims that observations irrelevant. That seemingly disproves the theory because, I mean, you shouldn't be married to your theories, but
Right.
that's not how people work. Science is a lie. Anyway, that's a little bit on epigenetics. And we can pause here before we talk about horizontal gene transfer, which we'll save for part 3 and the exciting conclusion of
Yes. I look forward to it, Ilya. This has been great. Thank you so much. And we'll see you all in part 3.